Welcome to the current state of stem cell and gene therapies for Parkinson's disease webinar. Thank you very much for our presenter, Amber Van Laar, who is a movement disorder specialist neurologist. She is the Senior Vice President of Global Clinical Development with AskBio. It's a biopharmaceutical company, and she's been a wonderful partner of Parkinson Foundation, Western Pennsylvania. And please take it away, Dr. Amber Van Laar. We're excited about this topic.
Yeah, no, thank you. I'm also, as you probably most people coming to this probably know that I'm very excited about this topic too. But I haven't really talked as much about the cell therapy side, but usually when we're talking about advanced therapies, cell and gene tend to kind of go together, and we have same meetings, that sort of thing. And a lot of the approaches to this are very similar. So I wanted to kind of lump these together. And I think there's a lot of questions that come up on the cell therapies too that I'm hoping I can maybe discuss with you guys today. So these are my disclosures, just to make it very clear, as with Casey already mentioned, I'm an employee of AskBio. So this is a gene therapy company. I'm going to be talking about some of the studies that we've had going. So just know that these are my disclosures here. Happy to talk about any of this more if you had any questions. So I think the first place I like to start this kind of discussion is, why do we even want to think about something like a cell or gene therapy for Parkinson's? This is a big investment. It's, a lot of work, and it's, dangerous in some cases, and it's, can be very sci-fi sounding. Why would we even think about doing something like this? And one of the big reasons, and I would say most of the programs we're going to talk about today, the reason we're even thinking about going after the Parkinson's in this way is the question of, can we actually change or slow down or stop the progression of PD? This has been the holy grail, for Parkinson's and other neurodegenerative diseases. And, I think a lot of us feel very strongly that we have to fundamentally do something very different to really tackle the ongoing progression of PD. And this is where a lot of the cell and gene therapies are being developed, is really to address that specifically. But beyond just the disease progression and looking for this holy grail, a lot of people, there's still a lot of symptoms and issues that really don't get treated adequately with medications on hand. And so is there a way that maybe we could prevent them from even coming on? Now, especially some of the non-motor symptoms that can really plague people for years and years before they even have a diagnosis. Is there a way that we can stave them off, slow them down, prevent them to even showing up or getting worse? Similarly, similar issues with more motor stuff, so more falls and freezing, a lot of other problems that maybe, again, aren't addressed well with medication. Could we potentially, avoid this, like falls from coming on board? Or is there a way to prevent this from happening in the first place? Again, symptoms that are not well treated with medications we have already. And then on top of that, a lot of people, it's many of you here know, if you live with Parkinson's long enough, the predictability goes away. And so can we maybe do that? Can we make it, more consistent, predictable disease, and in preventing the onset of fluctuations and other complications that tend to come up in people living with PD for a long time? So again, these are probably no big surprises here, but these are big issues that we haven't really addressed well yet. This is why we're thinking about something as big as cell and gene therapy. So what makes them different? Why are we even having this conversation right now? And I think a lot of it, and what we're going to be talking about today, is it's done differently. We have unique routes of delivery. In a lot of cases, what you're going to see today is they're delivering right into the brain itself. That alone is different and makes it a little bit more complicated. And I think it increases the level of fear a little bit too. And so I hope we have a chance to talk about some of that today. But unlike other diseases, there is a precedent here for Parkinson's. So we have something like the deep brain stimulation. So there's already a surgery that requires direct administration of these electrodes into the brain. But again, brain surgery is not for the weak of heart. So this is one thing that really sets cell and gene apart. But as I mentioned earlier, one other big difference is that we're really looking to alter disease course. We want to see if we can actually impact disease progression. So to really treat the underlying causes of the disease and not just symptoms. And so for what I'm going to be talking about today, really focusing on cell and gene therapies that are available or current or will be coming up, and really focused it here, what we have here in the United States. There's a lot of activity in the cell space, particularly outside of the US. But I just for the purposes of the group here, I think it made sense to really focus in on the US. So I'm going to start here with the cell therapies, and I think it's important to, again, kind of step back a little bit. Well, why cell therapy for PD? And I think this is actually more obvious than thinking about a gene therapy approach. You know, if cells are dying, we know in Parkinson's that there are certain types of brain cells that are dying. And the thought then is, why can't we just replace those cells back? And, you know, and then voila, it'll all be taken care of. And that's really been the way that this has been working for many years. Cell therapy for PD has been ongoing for a couple decades actually. And so right now, in the way that Parkinson's works is this this purple line here, this pars compactus, where the majority of those dopamine neurons, this is where they live. And these are the cells that are dying. And so again, this is the place you would want to put your new cells to make them work kind of like they would if they were native and if they were the same ones that you were born with. And in a lot of cases, what cell therapies are doing is making new cells that basically work like little dopamine pumps. So they are basically doing the job of levodopa without you needing to take levodopa. So again, this is where a lot of the cell therapy approach for PD has gone. But it's really we haven't seen this more prime time to date. And there's a few more things we'll talk about here coming up, but it's what are we replacing? Where are we replacing? And so again, we know that the cells are really dying. The dopamine cells that really cause a lot of the Parkinson's symptoms are in the substantia nigra. We also know that they're just uniquely vulnerable just because of Parkinson's. And this is, again, kind of where they sit in the picture that I was showing you before. So it's pretty low down here in an older part of the brain. That's the substantia nigra. Their cell bodies sit here, and then they project these long axons into the putamen. And so when you don't have these cells, they die. We're not sending as much dopamine up to the putamen. And when you don't have as much dopamine, this is what causes a lot of the symptoms that we see in Parkinson's. So the tremor and stiffness and slowness. And when our Parkinson's patients take levodopa, that is basically replacing that dopamine that is not being made because you're losing the cells that are here in the substantia nigra. Again, less dopamine, this is what brings on a lot of the symptoms that we know so well in Parkinson's. But it's really the putamen that's I like to call the business end of dopamine. So this is where dopamine shows up, and where it's acted upon is here in the putamen. And so it turns out that a lot of our cell and gene therapies are actually going after the putamen. So replacing cells here for majority of what I'm going to be talking about today. So there's some benefit to this. Again, if these are cells that are made to making dopamine, they're pumping it right into the part of the brain that can action, on that dopamine is the idea here. It's also a much bigger target. It's easier to get to. It's a little bit tight here in the brainstem. So this is why it can be dangerous to try and go this route. And yet I think it makes a lot of sense to try to go to the putamen. So just know that is a big part of the thinking behind why we would go to this part of the brain. So let's take one more step back and just talk about stem cells just more generally. So I think many people may have some ideas of what is a stem cell, but really basically it's the raw material. So we have, these very early cells when an embryo is starting to form, you get these cells that can basically turn into anything. And in fact, these are the cells that then make up over the 200 different cell types that we have in the human body. So, the cell can turn into a blood cell, a fat cell, a muscle cell. And so again, it has all that potential, within that stem cell. Now, there are other ways for us to get stem cells. And, one example is actually taking like a fat cell and turning the clock back to make it look more like a stem cell, and then essentially forcing it in a lab setting into a dopamine neuron. So there's two different routes that we can use to get stem cells for Parkinson's patients. Now, this is maybe a little more technical, but I think it's important for us to talk about exactly where we're getting these cells, because every study does something a little bit differently. And this also is a source of the ethical sort of, I think, concerns that come when we talk about cell therapies. So this is the different places that stem cells for Parkinson's have been looked at. So one is allografts. or excuse me, autografts. These are taken from self. There have been, in fact, adrenal glands that have been harvested from people and been used as stem cells there that way and converted into dopamine cells. There's also allografts, so it's coming from other humans that are just not yourself. So, for example, we can take fetal dopamine neurons. This has been looked at before too. But even the retina, believe it or not, has some dopamine cells that can be used. There's also xenografts. This has also been tried in Parkinson's. For example, there's using dopamine cells from pigs and then transplanting that into humans. So again, these have been tried. They all have their upsides and downsides, but this is just a smattering of cells where we can take, already cells that are producing dopamine and try to then replace them into a Parkinson's, a brain that's under the ravages of Parkinson's. Now, another way of doing this, and I think most people may automatically think about when they're thinking about stem cells, so we can take straight up embryonic stem cells, force those to then convert into dopamine cell. But there's also something called an iPSC. So this is an induced pluripotent stem cell. These are stem cells that are coming from an adult. They can even come from skin. And again, they're forced back into this early state that can then be turned into a dopamine cell. So again, lots of places we can get them. Most of these have been looked at some point in our history. So interestingly enough, there's actually been a lot that's been done so far for PD. Unfortunately, there is still, in spite of all this work, decades of work that have led us to the studies I'm about to show you here, but we're still asking ourselves, what is the right dose? How many cells need to be put in? What's the right type of cell? How, if I do get them in, how do I make sure that they're connecting to the right partners so that they work properly? Maybe there's the right type of PD patient that should be involved in these studies. Should we have them early? Should we have them later? But we still really are trying to figure this out right now. And then ethically, how does that make sense? Yeah, another big issue is how do we make sure that we get living cells that are accurately placed and that we can reproducibly do this in patient after patient after patient? This is also another big challenge that we're facing right now in this field. And another topic that we can get into if you're interested, but there's right now, this is basically like an organ transplant. And because of that, a lot of these studies require patients to be on immune suppression. So this can be everything from steroids to, more specific, harder, let's say bigger drugs, if you will, to really, really suppress the immune system. So again, these are a lot of questions that we're still grappling with in the field. So I'm going to turn now to clinical trials that are currently ongoing or soon will be upcoming with stem cells. So the first one I'm going to mention is with Bluerock. So this made a bunch of news earlier this year. I should also mention that they are affiliated with my company. And I'm going to talk about their phase 1 study that was looking at embryonic stem cells for Parkinson's. And they were looking in a more advanced stage Parkinson's patient for this study. So they recently conducted a phase 1 study. So this was open label, meaning that everybody knew that what they were getting. This was also looking at a dose escalation, meaning that patients with multiple amounts of cells were looked at. So they looked at 2 different dose levels of cells. To deliver the cells, this is done with a neurosurgical administration, so a brain surgery, and it's only one time. The cells that were used here were embryonically derived dopamine neurons. Now, some upsides to this type of approach. So this is directly replacing the cells that are dying in Parkinson's. The other thought here is that these are dopamine cells that are making dopamine. Therefore, we should be seeing relief of dopamine responsive symptoms. And in fact, they did see this. And I think some of the watch outs, and I'm going to kind of keep the same sort of structure here for all the studies I'll be talking about. There's some upsides and things just to kind of keep an eye on. So how do we know that those cells are surviving? It's really hard to show that. It's really hard to demonstrate that. But these are things that as the study goes on, we need to watch for. And I think the other thing to think about too is that this is another one of the studies that requires immune suppression. So is that causing any other side effects or that sort of thing? So again, keeping an eye on this one. They are getting ready for a phase 2 study, which they announced would start sometime this year. So I think this is probably one of the furthest along programs for stem cells here in the United States. They have reported some data out from this one. As I said, this is pretty far along compared to some of the other studies I'm going to show you. So it's some important call outs. So one, no major safety issues. In spite of being neurosurgically delivered with stem cells, with the immunosuppression, there's no major safety issues looking up to 18 months from the time of delivery. Another concern with stem cells, because this has been a pretty big problem in the past, was a problem with what we call runaway dyskinesias. So you get these wild, very difficult to manage dyskinesias with prior stem cell studies. They did not see that here, and that's a big deal, that they had a good safety profile when it comes to dyskinesias. And I think another call out here is they looked at immunosuppression, and there was no problem with when patients were on the steroids, and even after they came off, that there was no issues with that either. So overall, a pretty good safety profile. Looks like they were seeing some big improvements on some of the motor symptoms, and enough so that they're getting ready for a phase 2. So this is one of the big studies I wanted to bring up in the cell therapy world. I wanted to mention this one because this is another big study that's being performed. It's outside the United States, but this is one of the larger cell therapy studies that's being conducted in the UK and in Sweden. Also a phase 1 study that's open label, also requiring neurosurgical delivery, going against that larger putamen, sort of in the business end of the dopamine system. So you can see here, this is also a dose escalation. So they're increasing the number of cells that they're delivering. And it's in part because they don't know how many cells are going to survive and make it and then connect to other parts of the network. Now, one major upside and why I wanted to bring this up is that they are working with experienced centers. So these are groups and physicians that have worked with cell therapies and this type of population for quite a while. They're also looking at a more moderate Parkinson's patient. So this is sort of middle of the road type of Parkinson's patient. And just some watch outs is, again, this is a bigger study that's, been well funded. They're only taking participants from their observational study. They had opened up this observational study and then directly enrolled them then into this cell therapy study. And again, just as I mentioned before, some immune suppression is needed for this one as well. And just a note, we have the NCT numbers. These are the clinical trial numbers for all the studies that have them, and you can look them up here. Happy to share this with you as well. I wanted to mention there's several other studies, and if you go on to clinicaltrials.gov and you look up stem cells for Parkinson's, you're going to get a whole lot that shows up. So this is another one that I wanted to call out. So this is being done here in the United States, and this is using allogenic stem cells. So again, as I mentioned earlier, this is using from human. In this case, they're using adipose cells from a human that are then transformed into dopamine cells. This is being done through an infusion versus saline. So this is a controlled study. So not everyone's going to get active treatment, but it's multiple infusions over the 20 weeks. Now, this is an advantage because it's IV. That's really easy. And what they've done is they are trying to take fat cells from that person and then converting those cells and then giving your own cells back to you. So there's a big upside because there's a lot less concern for it, like an immune reaction when you're using your own cells. So some benefit there. Some things that worry me though, some watch outs that I would mention to the group here. There's no crossover, meaning that patients that had gone through this study and received just the saline, there's no plans right now on how to manage those participants. And then I think the other big thing that really worries me is, when you're placing brain cells, through an IV, how do we know they get to the brain? And if they do get there, how do we know that they're there and they're working and doing their job? So these are just, I'm probably gonna hear me say this about a few other ways, is trying to get those cells integrated into a network that's already been established, I think is really challenging. But I think particularly so if you're giving it IV and not right to the brain itself. And this is another interesting study, from Aspen Neuroscience. You can see the website here. So again, this is looking at autologous cells. So, same idea that you're taking participants' own cells and transforming those then into a dopamine cell. And what's really different about this group is they're using artificial intelligence to look at these cells specifically to pick which they feel are the best cells for transplantation. They've gotten a lot of news. There's fast track designation from the FDA. And actually, since I last talked about this, they've actually dosed their first participant earlier this year. So again, exciting story here with a kind of a different approach from a cell therapy standpoint, but active here in the US at just one single site. And you can see the fast track designation announcement that's here. And I just want to call out, there's just to make sure that everyone, I say this out loud too, you go on clinicaltrials.gov, you're going to find a lot of studies that are claiming to be cell therapy studies. There are also many, many other studies that are being created, conducted for cell therapy across the globe. And I just ask everyone, please be careful, be very thoughtful when looking at these websites and looking at these different groups that are doing these therapies. A lot of these organizations are not really being held to the same stringent standards that we would here in the United States. The other big concern is for medical tourism. This has been a big problem, I would say, particularly for cell therapy. People will, pay lots of money, go, across the globe to try to get cell therapies for Parkinson's. And I've seen personally some pretty bad outcomes from this. So I just would always please be very cautious. You should be only working with studies that have been listed on clinicaltrials.gov, and you shouldn't be paying to be part of a study. So just that's very clear. Just be very, very cautious and careful when looking at these kinds of studies. But here we are. Cell therapy has been looked at for decades for Parkinson's. So why are we being held up still? What's going on? And so one of the big issues is manufacturing. So as you'd seen earlier, with cells that are coming from yourself, they have to be, converted and transformed into dopamine cells. That takes a lot of work. And then how do you get them stored? How do you get them shipped to the place where they're going to be delivered? And then how on earth do you do that for millions of people living with PD across the world? So this is one major issue that's really held the cell therapy world back a little bit. But as I mentioned earlier, again, we still don't know what's the best type of cell. Is there a better type of way of delivering this? Do we need to do direct delivery to the brain? Can we do IV? We still don't know that. And again, the ethical concerns for cell therapies will be there for a while. And so this is a non-starter for some people, and for others, not so much. But this is always going to be there for now. And some of the other issues we brought up already. So this potential need to use medications to suppress the immune system. It's which ones, for how long do we need to do for everybody? So that's another big issue that is being actively looked at right now. And as I mentioned earlier, this concern for runaway dyskinesias, this had been a big problem and really kind of, I would say, took the field back a little bit from a cell therapy standpoint. But I'd say there's some good news seeing the most recent data coming out where this hadn't happened before. I think we learned, but this is something that we still need to keep our eyes on because it had been a big issue in the past. And again, I mentioned this before, this is going to be an issue for every cell therapy that's out there right now, is how do you prove to yourselves and to regulators and to patients and their families that you've done what you said you've done? How do we know that we have transplanted successfully cells that are alive and working properly and have integrated themselves into the brain? It's really not an easy thing to demonstrate. So again, these are all items that have slowed things up. And I think it's important for all of you here to know what's going on too. It's not easy to get these studies done. And just last thing on the cell therapy, this was an interesting survey that was conducted by the Michael J. Fox Foundation. And I would just have you focus on the top three here. So by far and away, most people who responded here said that they would think about enroll in or have actually already participated in a stem cell therapy program. So I think, I found out that it actually surprised MJFF that a lot of people, the majority here had more interest in cell therapy. But this is just more reason for us to have more conversations about this. And I would also ask you all here, think about yourself. How would you answer this survey? I think you might see probably a similar type of distribution here. So I wanted to pause here and see if there were any questions on cell therapies specifically. I don't know if anything came in through the chat.
I didn't see anything come through the chat.
Okay.
If you any questions, or there will be time at the end if not.
I'm happy to keep going. All right. Oops. Okay. So I'm going to switch over to gene therapies. Hopefully some of you are familiar from me talking about this in the past, but I'm going to still do some basics of gene therapy one more time. So this is Gene Therapy 101. With gene therapy, we're taking advantage of what your cells do every day anyway. And really kind of starting at the beginning here, when we talk about genes and gene therapy, we're talking about DNA. And DNA is the blueprint for all the things that our cells need to do and make. Cells just on their own, we do this every day. The cells read the genes, and then from that instruction from the DNA, they then make proteins. And the proteins are really the business end of a cell. So this is what helps keep the cell alive, or makes our eyes the color that they are, makes our muscles work. So proteins are really important. But they're all made based off of the template of our DNA or the gene. Gene therapy can be harnessed in a couple of different ways. So we can either deliver new genetic materials, we can deliver a new gene of something that's maybe already there, but we can make more of that protein that we may want. We can turn off a gene that's bad. It's been mutated, it's not making a right type of protein from it. We can turn it off. We can also replace a bad copy of a gene and replace it with something that's more healthy. But for Parkinson's gene therapy, and a lot of I'll be focusing on today is really this idea that we're going to introduce another copy of a good gene. Now, the other part of trying to understand gene therapy is how we package and deliver our gene therapy. For AAV gene therapy, we're using AAV. So this is this vector here. So it's basically a viral vector that's been emptied out. So it's been altered in a way that it can't make humans sick. And so AAV, for sure, it cannot make you sick, but it has all the outside properties of a virus. And in our case, what we're using the AAV vector for is like an envelope. It's one way for us to deliver a message to the cells of the body that we want, while carrying the right message, or our DNA or gene that we do want. So kind of going back to this analogy, I'm hoping some people are familiar with this, but we think of our gene or our piece of DNA here as like a letter. So it's a message that you want to deliver. And then we think of our vector, or this AAV, like an envelope. And just like with a letter and envelope, you need to put them together. You need to put your letter and package it up into the envelope. And that's exactly how we do this here. So we put our gene into our AAV. But now you've got this posted, it's ready to go. You need to get it delivered. And in our case, we're using delivery right into the brain, kind of like a delivery truck. So we're putting it into the right neighborhood, if you will. And then the gene therapy itself, so we now have our gene inside our AAV, does what it knows how to do. It's able to then deposit and leave our gene, just our gene of interest, into the cells that we want. And in our case, this would be a brain cell. So we have the gene going right into our brain cell, and the brain cell knows and does exactly what it's supposed to do, because it does it every day. It reads the instructions, and then it makes the protein that we've asked it. And this can be all kinds of different proteins. But the protein basically then works as the medicine. So we start all the way here with just a little piece of DNA. And really what we're trying to do is make more of a protein of interest that we think is going to be therapeutic or act like a medicine. As you may be familiar, I've been working on this AAV2 GDNF gene therapy for Parkinson's for quite some time. And I think I'll focus there first for gene therapy. So again, everyone here, I'm sure, very well aware, when we lose in a healthy situation, when we have lots of dopamine, that's these red dots here, we have lots of dopamine floating around because we have a healthy dopamine cell that's releasing it. It gets picked up, and this is how you get normal movement and, no shaking, nice, smooth, fast movements. But when you lose dopamine and it's just not able to hit on these receptors, this is what gives the stiffness and slowness and tremor. Now, with GDNF, gene therapy, the idea is that GDNF itself is a growth factor. So it's called a trophic factor. It's like food for dopamine neurons specifically, and it really helps promote the overall health and ability to function of these dopamine neurons. In fact, when we're young and actually still in utero, we have really high levels of GDNF. As we get older, those levels reduce over time, but you still need a little bit as an adult. The hope here is that by bringing back more GDNF, so we have that little bit of GDNF as a gene, we make more GDNF, that will actually help to restore some of the damage that's been done by Parkinson's. The idea is that with GDNF around, you can convert this kind of sick, more pruned dopamine neuron and convert it into a healthier neuron with more sprouting, so that it has more ability to make and then respond to the dopamine that's there. The idea is, again, that GDNF can help support these sick but not yet dead neurons that are remaining in the Parkinson's brain, and really help us to bring back more normal dopamine production and response, to really mitigate especially those movement symptoms in particular. As with a lot of the other therapies we've talked about today, this is also delivered directly to the brain. This is done in a slightly different way from a lot of what I've shown you before, in that we're coming through the back end, coming through the back of the head, and then injecting very slowly into the putamen, as we talked about earlier. This is actually a technique that's been used for over 15 years for Parkinson's, but a few other diseases as well. And it's only a one-time procedure, which I think is a big deal. If you're going to be doing something as serious as brain surgery, it's better to have it only done once. And I think the other advantages to this kind of approach is that there's really nothing left behind except for the gene that we want. So there's no hardware, there's nothing that needs to get worked on or tweaked over time. This is really a one and done procedure with close monitoring afterwards. And so we do think that GDNF, again, this is based off of decades of work in animals, both rodents and primates. That there's this ability of GDNF to bring back those dopamine neuron, so not really growing new neurons, but making those cells that are still there, making them work better and healthier. We know that loss of GDNF is part of the problem that leads to the continued progression and loss of dopamine cells in Parkinson's. And we do hope, and this is the study that we're looking at right now, is can GDNF support those cells, help them grow and stay and live longer? Can we slow down or even potentially reverse disease progression? Those are the goals with GDNF. So this is a study that is active, no longer recruiting. The study is done with new participants. But this is the second study looking at the same exact drug, so this AB1005. This was open label, meaning that everybody knew that they were getting active treatment. This is a one-time single dose infusion going again into the putamen, as you can see here. Unlike a lot of the other programs I just showed you, this is done in the MRI machine, so we can see exactly where we're going and where it's not going with the gene therapy. So you can see this black line here is actually one of the cannulae. So it's basically a long, thin tube that gets inserted into the brain very slowly. The white you see coming in here is the gene therapy itself that we can see in the MRI. What you're seeing in this video here is actually several MRI images that have been stitched together into a video to give you a sense of time. This actually takes hours and hours and hours to do. It's just been sped up so you can see sort of the growth and how this expands. The idea here is that we want to fill up this gray comma shaped structure here, which is the putamen, and only the putamen. And then we feel that we have a better chance of doing that with being able to look at the infusion in real time in the MRI. So again, some upsides here. This is a unique approach. GDNF has been looked at for decades for Parkinson's, again, with this ability to potentially regenerate and cause, lead to sprouting of the immune system. And the potential to relieve dopamine responsive symptoms like tremor and stiffness and slowness. And again, like the others, this is a one-time procedure. Now, some things to worry about with this one, some watch outs here too. It's a long procedure. To do this, the surgery takes quite a bit of time. And I would also argue there's been a lot of problems with GDNF in the past. We just haven't seen the potential of GDNF really come forward yet. And I could talk about that for hours, but I think this is some of the skepticism we hear in this field too. Now, the study went well, and in fact, there's a phase 2 study that is now active and looking for participants now. So if you have any questions about that, please let me know. But I do want to give you some takeaways, though. Again, this phase 1b is wrapping up at this point. But so what did we find? I think the number one thing I'd like to start with: no major safety issues. This is always a concern, especially with the neurosurgical approach. But this was looking at 18 months and even beyond. The surgery was well tolerated, and patients are doing well. Now, looking at 18 months post treatment, we were looking at two different groups. So, looking at a cohort with mild Parkinson's. So, on average, this was a group of people about 2 years from diagnosis, versus more moderate Parkinson's, which was about 8 years from diagnosis. And in the mild group, they were doing quite well. They continued to do quite well, and they actually looked very stable over this 18 months and beyond, actually. Whereas our moderate group, who were doing a lot worse at the time that they came into the study, demonstrated pretty significant improvement in a lot of motor domains, especially. Either way, this study was also very encouraging, and this is what's led and why there's now a phase 2 that's open and actively recruiting right now. Now, one big difference is this phase 2 will be a blinded study. This is going to be really important to understand, what GDNF is doing and not what the placebo effect is doing. And so this is a really important study for the entire field. So let me know if you have any questions on that. Now, I wanted to dive into a few other gene therapy studies. There's actually not a whole lot that's going on right now in Parkinson's, and not a whole lot going on in the United States. So not too many to dig into here. But this is one that I've been watching for many years now. So AAV-GAD for Parkinson's. There was an early study done many years ago that it looked interesting and kind of just got really quiet. They've since restarted, and now we have this phase 1/2 that's ongoing. Again, very similar, one-time surgical delivery. The one difference here is that this group is going into the subthalamic nucleus, the STM, and using this approach in a very different way. So this idea is trying to use this GAD to change the way that the circuitry is talking to each other. And the way that this is coming about is that there is potential for disease modification. So it's a very, very different approach than anything else we've talked about already today. There is some historical experience here looking at this in the past. This study is interesting. So some of its upsides is that it's open to a lot of PD patients, where some of the other groups are looking at a very narrow slice of Parkinson's. And I think it's really interesting that they have a very unique mechanism of action, trying to look to normalize the electrical activity in between these areas to help alleviate Parkinson's symptoms. Some watch outs for this one. So there's a sham surgery because this is a blinded study. So we have some participants who are not going to know if they got active treatment or not. And it's also not known, from what I could see, if there's crossover, meaning are participants that received the sham, will they be eligible then for active treatment later? I mean, these are things as a patient I would want to know. So this is one to keep an eye on here as well. This is another study that's been going on for some time. So this is really focusing on a type of Parkinson's where there's a known mutation in GBA. So this is a phase 1/2 study. They're also looking at dose escalation. So what's the right dose here? The one difference here is they have this sterna magna infusion. So what's that? So this is not going straight into the brain. This is going into this tiny space here that's fluid filled with CSF, so the cerebrospinal fluid. So this idea that you can get this little pocket of fluid, deliver your gene therapy there, and then it will circulate through the fluid around the brain. So another unique aspect to the studies, they're looking for advanced Parkinson's. So we're talking about patients with Hoehn-Yahr 3 to 4, meaning that this is probably a lot of balance issues, this sort of thing, difficulty walking around for sure. So, this is some of the upsides here. So we're replacing an enzyme that's, not working well in these particular patients with a known mutation in this GBA. Another advantage is it's not going straight into the brain. This is going into the fluid area, which is, a lot easier, still a procedure, but not directly into the brain itself. And it's open label, meaning that everybody going into the study will know that they're getting active treatment. Now, some things to watch out for in this one. There is still immune suppression that's needed for this particular gene therapy, and they're only looking for patients with a GBA mutation. But I would argue there are some patients that probably are not eligible for a lot of other studies because of the GBA mutation. And so if you know of anyone that has a GBA mutation, this is a great study to think about looking into. This is where my big disclaimer here. Be careful where you're getting your information. Please talk to your neurologist or any physician that'll hear you out. There's just, it's so easy to go down the wrong path on some of these therapies, and a lot of people try and take advantage. Other websites I like, the Michael J. Fox website, they do a good job of, especially the Fox Trial Finder, giving patients really direct information on where to find Parkinson studies in particular. You'll hear me say it a bazillion times. I'll say it again. Go to clinicaltrials.gov. It is not the easiest website to use, but any legitimate clinical study is required to be listed on clinicaltrials.gov. So always check if you're interested or thinking about a clinical study, always check there as well. But also go look at other companies. If you heard of something, go look it up. There's a lot of universities. There are some of these cell therapy studies in particular are only being done at like one location. So you can dig into what the university itself is talking about with regards to the therapy. And I'll say it again, talk to your neurologist. You just got to talk because these are, especially when we're talking about a surgical approach for these types of therapies, this is something that it shouldn't be taken lightly. You need to talk to your family, talk to your neurologist, and everyone around. So please look to learn more. And then I'll just throw in for AskBio as well. We're very interested in keeping close tabs with our patient communities. I'm here, and you've seen a lot of other people from my organization. We are very keen to keep the patient voice right at the forefront of everything that we do. And with that, I'm going to say thank you. And look, I hope we have a few questions. I'm open to anything.
Thank you, Dr. Van Laar. Yes, there are some questions. Would you like me to read them to you?
If you don't mind, because I cannot see them.
Yes. Okay. Well, the first one is a comment. To ask anywhere near an intelligent question, I'm going to have to rewatch this a bunch of times. I agree. Easy to understand as the presenter. You are excellent. It's just a lot to absorb.
It is. Yep.
Yes. Thank you. In the delivery to the pitamen? Oh,
yep.
pitamen, yes.
Yep.
Is there any downstream positive brain improvement? I think that's the correct question. Kathy, is that correct? Is there any downstream positive brain improvement?
So let me see there's probably two ways. So sometimes in like DBS, we will get like a bit of improvement because we've placed an electrode there. That's usually when we go to a certain part of the brain called the STN, so not really putamen per se. So no, I don't expect, and usually if we do get something like that, it's really quick. It doesn't last very long. The other thing that I get asked a lot is, does it cause any permanent damage when you're passing something into the putamen? And so, in fact, it's something that we're looking and watching for very closely. This is even with DBS, like this is, a concern as well. Now, some of where this becomes more of an issue is this is why we have to go slow. The brain tissue itself, and again, because of the way that the devices are being made, it really does help to push the cells aside. And so there really isn't, if you just jammed in there, yeah, that would be a lot of damage. But just trying to do it very slowly and gently and watching, with the MRI, we're able to do this fairly safely. But yeah, there's a scar that can be left behind just passing anything through the brain. There can be a scar. And what are the implications of that down the road? We're trying to figure that out now.
Thank you. In your experience, what are the factors that most discourage people from participating in clinical trials? And can there be ways to reduce them?
Yeah, no, I really appreciate that because I think this is, I mean, it's hard enough to get people into clinical studies, but into a neurosurgical clinical study where you might get, you might be blinded and not know if you received anything. I mean, that's a huge ask. I mean, that's asking for, the world and asking put people, people's lives in our hands. And that's not lost on me. Um, and this is, I have to ask myself, would I do it? Right. I mean, I can't be asking other people and not even, think about how I would do this on my own. And it's scary. I think that it's risky, and especially for a phase 1 study, if it's never been done before. And what we see is, women tend to be a little more risk adverse. And so we tend to get more men, especially in these earlier clinical studies, especially if it's neurosurgery involved. And so I hope that it's a lot of like educating, being out there, talking to people, trying to dispel these fears. But there's the just neurosurgery, that's a non-starter for a lot of people. But I would say, even if it's not this kind of study, just getting involved in something would be wonderful. Even family members can participate in some studies as a control, just because we don't learn anything if we don't have participation in these studies. So if you have any questions on that, please reach out to me, because I know it's scary to make that leap, but we won't be able to make any headway in this horrible disease without you.
Thank you. And thank you this is a comment and then a question. Thank you for forging ahead with phase 2. I think your study has potential to help several diseases, which is very exciting.
Yeah, me too.
And how is the spinal fluid assessment working? Is it still showing success in finding misfolded proteins?
So I might need someone to clarify it for me. So is this the seeding amplification assay? Anyone know?
And this is Carol asking the question. Carol, oh, there you go. Yay.
There I am. Yes, that's what I was asking about. Also from the Alzheimer's perspective, because I think it's an exciting test.
Yeah. Yeah, so the seeding amplification assay, just as a background. So this is a way for us to detect and diagnose. This is the goal, is to diagnose Parkinson's, and ideally diagnose it before you have symptoms, so that maybe we can start doing something before things get out of hand, right? So this has been put forth by the Michael J. Fox Foundation. And the other way that the seeding amplification assay, or SAA, is being looked at is, can we distinguish MSA from Parkinson's, from DLB, from PSP? That's the other big mission that's going on right now. So the other way that they're using this information is, if we can, in fact, identify people very early on, what types of therapy should we be using in that kind of situation. And so that's actually what they're doing right now. And I think it's quite fascinating, working directly with a lot of sponsors that may have drugs or something that's already going that may stop Parkinson's in its tracks. So that's what they're trying to apply it that way now. And the other goal is can we do this outside of CSF? Can we do this in blood or some other fluid that's easier to get to than spinal fluid? So that would be another big mission of theirs. There's another alpha-synuclein assay, like in skin biopsies, that's also gaining a lot of traction. I think they may have spoken to the team here too. So there's quite a bit now. And what you alluded to, Carol, is it's kind of like, is it like Alzheimer's? And in fact, that is exactly where they're going with this. They want to convert this to a biologic diagnosis of PD. You no longer have to wait to show a tremor or have the bradykinesia. They're going to say, you've got this in your seeding amplification assay, and you've got this sort of brain, imaging. You've got Parkinson's before you may even have symptoms that show up. So that is where we're headed. That's definitely where Alzheimer's is going. This is Parkinson's is right behind what they're doing with Alzheimer's too.
Fantastic. Thank you.
Yeah, thank you.
Any additional questions? There's a thank you. If you are able to raise your hand, unmute yourself and ask the question that way, you can do that as well.
Yes, please.
Dr. Van Laar, can you speak about today? Today is the Parkinson's National Day of Action. Any comments, thoughts?
Yeah, I feel like there's momentum. I feel like, I actually was on the Hill not that long ago. We'll see if this really gets us some headway. I have a lot of hope with this one. Just there's been a lot of efforts in the past to get something similar going. And I really am hoping that we're actually taking some of the steam from the Alzheimer's world. They've had some successes here. Can we take from that, learn from that, and move forward? And in fact, that's part of what I see as a benefit with this is a knock-on effect from the Alzheimer's world. So let's see how it goes today. I'm really excited. I have a lot of hope that this is going to make some great waves for the Parkinson's community.
Thank you. And there's an email in your inbox that Lauren sent from Parkinson Foundation Western PA yesterday. You can sign the petition. You can send an email to Congress if you choose to do so. It's a big day. And Lauren included the link in the chat.
Yeah, please sign up. Let your voices be heard.
Absolutely. Any last questions?
Hi, this is Christine.
Oh, there's Christine.
I just wanted to alert people to look for the NBC story about Parkinson's disease that features Marilyn Raimondi. And also look for the Viewpoint with Dennis Quaid video that's going to be all over the TV nationally in October.
Can't wait. I hope to see some people.
Muted.
Yes.
I hope to see some people at the Step Forward walk as well. If you have any questions about this, you are welcome to ask me then too.
Right. Oh, look, there's one last question. I believe you stated the GDNF phase 2 was going on. How long will it take to go to phase 3, where people who have PD will have access to the therapy and not be getting a placebo?
So phase 3 is quite a ways off. We need to focus on the phase 2. And it just started. So I think it's almost impossible to find that crystal ball to let me know when phase 3 starts. But phase 2 is open. It's actively going.
And certainly if you have questions directly related to the research, ask first at askbio.com is the email.
Yes, please.
We hope to see you at Step Forward.
And a shameless little plug for Step Forward.
Thank you, Dr. Van Laar. And
My
you
pleasure.
can watch this again on our YouTube channel if you might need to absorb some more of the information.
Appreciate everybody. Thank you so much for the time.
Thank you so much, Dr. Van Laar, and thank you everyone for attending. Until next time.
Till next one. Bye, everyone.
Take some action. Bye.